Hi, my name is Monica, and in this video I will explain how to label and purify proteins with Alexa dye and HS ester for our experiments on Fluidity-1W or Fluidity-1W serum. In this demonstration, we will be labeling SARS-CoV-2 receptor binding domain as described in our user guide. When labeling your protein, there are three main steps in the process. First, the labeling reaction itself, then the post-reaction purification of your labeled protein, and finally, characterization of the labeled protein. To carry out a labeling reaction, you will need the material shown here. Ideally, your protein around 1 mg per ml, and definitely in an amine-free buffer. If the buffer your protein is in contains free amines, such as trees or amino acids, it will need to be first exchanged into an amine-free buffer. To do this, follow the protocol for post-reaction purification to perform buffer exchange. You will also need Alexa NHS dye, anhydrous DMSO, labeling buffer, which is 200 millimolar sodium bicarbonate of pH 8.2, pipettes and tips, and an Eppendorf tube per sample being labeled. If it's not already in solution, dissolve 1 mg of Alexa dye in 80 microliters of anhydrous DMSO to obtain 10 millimolar Alexa stock solution. If you are using an existing solution, warm the dye solution to room temperature before opening it to avoid condensation. This has been pre-made for the video. Transfer 100 microgram of your protein into a new Eppendorf. For our 1 mg per milliliter receptor binding domain or RBD sample, that is 100 microliters. Add 20 microliter of bicarbonate labeling buffer and mix by gently pipetting up and down. Try to avoid forming bubbles and do not vortex. Add 1.5 microliter of Alexa dye stock solution to the protein solution and pipette up and down 5 times gently using a 100 microliter pipette to mix the reaction. Protect the reaction from light and incubate it at 4 degrees overnight. If you are labeling a protein other than RBD or a different amount of protein, you will need to adjust the volume of dye used. We have a calculator on our website that can determine the volume you need to use. Just click on the link below this video. After overnight reaction, the chemical labeling will have taken place and it is now time to purify the labeled protein away from the free dye. To do this, we'll use a 1 ml Pierce desalting cartridge and the connector that comes with it. We'll also need a 5 ml and a 1 ml syringe, a needle and 10 microtubes. Finally, we also need a buffer to transfer the protein into. We routinely use PBS, but if your protein requires the use of a different buffer, then use that instead. At this point, free amines are no longer a problem in the buffer, so can be included if required. Before starting, open your microtubes and place them in a rack. To prepare the column for purification, snap the end of the column and replace it with the provided threaded cap. Remove the threaded stopper and replace with the Fluor adapter provided with the cartridges. Fill a 5 ml syringe with your elution buffer and carefully remove air bubbles. Injecting air onto the column should be avoided. Insert the syringe tip into the adapter, remove the cap and flow the buffer through the column into a waste collection tube. The target flow rate is about 1 milliliter per minute, so this should take about 5 minutes to do. Once the column is prepared, it is time to load the sample onto the column. To do this, first draw up to 200 microliters of your labeling reaction into a 1 milliliter syringe using a needle to reach the bottom of the tube, then remove the needle. Carefully remove air bubbles from the end of the syringe, insert the tip of the syringe into a Fluor adapter on the cartridge, and push the sample onto the cartridge, again taking care not to load any air. The flow through from loading also goes into a waste tube. Disconnect the syringe and dispose. To elute your protein from the column, fill a fresh 1 ml syringe with elution buffer. Remove air and insert it into a Fluor adapter. Elute the column at the rate of about 1 ml per minute, collecting into the racked microtubes. Collect about 100 microliters in each tube. The volume markers on the syringe are very helpful in monitoring the volume dispensed. When the 1 ml is consumed, the syringe can be disposed of. Cap the cartridge to prevent it drying out while you characterize your purified protein. The column can be reused for more of the same protein by washing with 5 ml of water or buffer before loading the next sample. Measure the absorbance of each fraction at 280 nanometer and the absorbance wavelength of your dye. The protein is expected to elute in fractions 2 to 4. This can be verified by a nanodrop measurement and the amount of protein and degree of labeling determined. The late eluting fractions will show dye absorbance but not protein absorbance as they contain free dye. Pool protein-containing fractions as appropriate. We recommend measuring the hydrodynamic radius of your purified labeled protein on your Fluidity-1W or Fluidity-1W serum instrument to confirm that all free dye has been removed. If the size measured is smaller than expected, it is likely that the sample still contains free dye and a repeat purification step is recommended. Thank you for watching this video. For more information, please visit our user guide. The link to this can be found below the video.